Okay, and I'm back. Welcome back to the Diaries of a Coronavirologist YouTube channel. It's been a couple of weeks since I posted. Apologies. I've had a few people asking if I was still going to be posting. Just needed a week off from doing these videos. Things piled up a little bit, got a bit stressful. Had to have a little weekend away, which was very restorative. And then I did record this video, which is coming up last week, but I wanted to sit on it for a little while and think about it and work out if there was anything I could explain with better clarity. So I'm doing another video on the lab origin hypothesis idea surrounding SARS2. And again, it's another long video like my last one. I highly recommend, if you're interested in this topic, going back and watching the last one because they really do feed into one another. They're both long, and I'll timestamp this one as I did with the last one so you can jump to certain things if you're interested in doing that. But if you're interested in this hypothesis that this virus could have come from a lab, I really want to go into it with a lot of detail and discuss it with as much clarity as possible, and that does require doing slightly longer videos. Anyway, with this little preamble said, I'll now lead into the video I recorded last week, so the dates will be a little bit off. And I'll get back to doing slightly shorter videos and move away from this topic for a little while unless anything massive comes to light, and then of course I'll approach it again. But hopefully by the end of the week I'll be posting another shorter video discussing more general things about SARS2 because there's been some interesting stuff this week and last week and the week before that I can very easily talk about. So without further ado, here is the video I recorded. I think it was on the 18th. I guess we'll find out in a second. Okay, welcome to video number 26 of the Diaries of a Coronavirologist YouTube channel. Today is the 18th of June, and we're up to about 8.4 million confirmed cases of COVID-19 and about 450,000 confirmed deaths around the world. So today I'm doing another video talking about the origin of the SARS2 coronavirus that causes COVID-19. I'm doing it slightly differently to what I had planned. So in my previous video, I laid out arguments for why I think this virus evolved in nature. And if you've not watched that, it's probably worth watching and then putting these two together because they're going to directly follow on. What I said in that previous video is I was going to take the comments and the feedback and look at them and discuss those in this video. I'm going to do that slightly differently today because my thinking on everything has evolved slightly. So I'm going to use a couple of the comments that helped generate some of my new thoughts and then talk about a new paper that I saw today that's also helping my thinking on this. So before going into that discussion, as a general point, I named this channel Diaries of a Coronavirologist because it's me on a day-to-day working as a coronavirologist dealing with this SARS2 pandemic and working with the virus in the lab and working extensively to try and find ways to combat it and talking about coronaviruses and trying to make things accessible to everyone because there's so much interest. Now, all of that, I'm approaching as a scientist. I'm looking at the data, taking what I know, building ideas, and I'm discussing those. As new data comes to light, I'm going to discuss it, and my thoughts may evolve and may change. And that's how science works. You take what you know, you take what's been shown, and you develop ideas, and as new data is provided, you change those ideas and you move forward and into new areas of thinking. So I'm not going to be dogmatic about anything. I'm not attempting to be dogmatic when I say that this virus, I think, was evolved in nature. And I stress I think because we don't have strong enough evidence to say it came from nature. And in my view, we don't have strong enough evidence to say it came from a lab either. So there is a degree to which my ideas and thoughts that I'm putting out are just speculation built upon the data. Anyway, with this video that I'm doing today, new data has been published and it's influenced my thinking, and I'm going to share that as part of the Diaries of a Coronavirologist aspect. So just before I go into the discussion of all the science, I want to try and summarize things into a small packet as best I can. Ultimately, I think there is not enough evidence to conclusively say this virus came from nature, and I do not think there is enough evidence to conclusively say that this virus was engineered in a lab and accidentally released. I think there's evidence enough to say it wasn't maliciously made, and maliciously released, but I don't think you can rule out either side of the argument of lab-made accidental release and nature. So therefore, it all becomes speculative. And where I land on that speculation is that this virus was something that evolved in nature. We know so little about the full genetic diversity of viruses that are out in nature. We've scratched the tip of the iceberg. That I think with all of that diversity that exists and the fact that genetic recombination can happen and all these sorts of things, I think this virus evolved in nature. Now, I'm going to lay out the arguments, or some arguments, for why this virus may have been something that was engineered in the lab. And when I try and strongman these arguments for myself and think about them, I can see a compelling case for why this virus emerged in the lab and how you could argue against everything I'm suggesting about it evolving in nature. But I think there's a big assumption that, at least when I'm thinking about it, I arrive at, which is that there has to be some degree of cover-up going on. And I don't want to make that accusation. I guess maybe being Pollyanna, I don't want to think that that's being done. I want to think that the science was being done honestly and sensibly and with true scientific rigor, and that combined with the thinking that there is this broad amount of genetic diversity we have no idea about leads me to a point of thinking this came from nature. So I'm not going to make accusations of a cover-up. That is not what I'm doing. I am just discussing both sides of the argument and trying to have an open, honest discussion about the science behind them and why my thinking has changed from the previous video somewhat. Now, with all that preamble said, let's get into the discussion. What I'm going to do is take two of the comments and questions that were put on my previous video and use them as a springboard to go into my discussion, a further discussion about whether this virus may have been generated in the lab and how my thinking has changed somewhat since 2 weeks ago when I made the last video. So the first of these comes from Keith Bell, which reads, completely unsatisfactory, all caps, discussion of the furin cleavage site, which is the smoking gun evidence for lab origin. Who are you to question why a scientist would add the furin cleavage site when we know it's already been done many, many times? So just before I get into the discussion of the specifics about the furin cleavage site here, I want to explain why it might be important, or why it is important. I quite often talk about the importance of the spike protein and the receptor. It's come up many times on this channel. I'm going to be talking about it again a lot as I go through this video tonight. So the spike protein on SARS2 binds to the ACE2 receptor, and this is essential for infection of any cells. The metaphor I always use is a lock and key. So the spike protein is the key that fits into the lock, which is ACE2. So that's just the first part of how a virus infects a cell. The SARS virus, when you think about those spheres with the spikes that you see in the pictures, that sphere is made up of fats and proteins, and within that is the viral genome. In order to get the viral genome into the cell to then infect that cell and make more copies of the virus, the fats surrounding the virus must fuse with the fats that line all of the cells, so lipids, to use the more correct term. So the lipids on the virus membrane must fuse with the lipids of the cellular membrane. And the way that fusion is done is through the spike protein changing its shape. So the way to use the same metaphor, the lock and key, to keep with that, it's like the key turning in the lock. So the spike protein binds to ACE2 and then undergoes a series of conformational shape changes that allow it to bring the two membranes close together to fuse and produce a pore which the genome can go through. Those conformational changes in spike that allow it to open the lock are pretty much all brought about through protease cleavage. So cellular enzymes, cellular proteases, chop up the spike protein at certain places, and this allows it to refold once it's chopped and cause that fusion event, which allows the genome to enter the cell. So the furin site is very interesting and potentially very important because furin is a cellular protease that can cleave spike and lead to those conformational changes that allow it to infect a cell. Now, there's a thought that if there's a furin cleavage site in the spike protein, this may allow for the virus to infect more cells than it otherwise would, because furin is found through a lot of the body. So it's found in a lot of places in the lungs, which may allow the virus to infect more. But this is somewhat hypothetical. In the lab, in cell culture, adding furin cleavage sites or removing them from viruses that have them can impact the level of infection of cells in a lab. But as I'll mention probably multiple times in this video, infecting cells in the lab is very different to infecting a full organism. So it's possible that the furin cleavage site may play a role in how easily this virus can infect cells, but it's somewhat unclear. So with that said about what the furin cleavage site might be important for, I'll now focus back in on Keith's question, just to go into a little bit more detail about the furin site in the case of SARS2 or in the case of coronaviruses as a whole. So I will admit I was wrong on the furin cleavage site, and I didn't pay it enough attention when I was piecing together my thinking for the last video. So to sort of lay out a little bit about where my thinking was at in a bit more detail, is that if you look at the human coronaviruses that were known before SARS2, there were 6, and these nicely divide. So of those that have a furin cleavage site, there is MERS, HKU1, and OC43. And of those that do not have a furin cleavage site, there is SARS1, 229E, and NL63. So in my view there, it suggests that the furin cleavage site is not a determining feature of virus spread. If you just take the seasonal coronaviruses, they all spread very well and cause common colds every year. And there's that split, 50-50 split between them. So there's nothing to suggest the furin cleavage site actually impacts transmissibility in a human, even though it may impact infection of cells in a lab, which are a bad model for humans, as I talked about in my previous video. Then if you take the pathogenic coronaviruses, MERS and SARS, SARS-1 spread a lot better than MERS. Within a matter of months, SARS-1 caused 8,000 cases, while MERS in 12 years has caused 2,500. So if anything, you would come to the conclusion that the furin cleavage site is not important for transmissibility and may even have a negative impact on it based on MERS. That thinking may well be slightly wrong though, and there's a lot of caveats to that about different receptor usage and all these kind of things, so I will admit to getting this one slightly wrong. Now the other part of my thinking on the furin cleavage site I definitely didn't go into enough detail on in the previous video. So I talked about recombination in the previous video, which is swapping of genetic material between viruses. The easiest explanation for this is with influenza that has 8 genetic segments, so 2 different viruses, each with their own 8 genetic segments, could infect the same cell, and then any kind of combination of 8 segments could produce a whole new virus. The same thing can happen with coronaviruses, albeit there's not different segments, it's just one single strand of RNA, but there can be recombination events that allow genetic material to swap. So where the furin cleavage site is in coronavirus, in SARS2 coronavirus, there is evidence that recombination events can occur at that site in other coronaviruses found in bats. So there is a bat virus called RMYN02, which has also had a recombination event at the equivalent site. That hasn't put in a furin cleavage site, but it has put in 2 amino acids. So recombination events can occur at that site in bats. Remember my earlier point about how little we know about genetic diversity of viruses out in nature. For me, the fact there's evidence that you can have this recombination event at that site is evidence that you can have a recombination event that led to the furin cleavage site. There's other viruses that have furin cleavage sites. I've just given you 3 examples of humans, let alone other ones out in other animals. So my thinking is that furin cleavage site isn't necessarily a smoking gun for lab origin. It is something that could evolve in nature. However, I'm using this as a springboard to go into the discussion about the lab origin more. So I'm going to come back to it and say why potentially it may have been used in a lab, because I am more open to this possibility with the more I've learned and with the way my thinking has changed. And just before moving on to the next question, since my credentials were raised, just for anyone who hasn't been watching these videos from the start, I'll just reiterate what I said in one of my early videos. I'm a PhD trained scientist. My PhD is in cell biology, where I study viral entry into cells and immune proteins that inhibit that. And I'm now a postdoc in a lab that studies coronaviruses, and I've been working on coronaviruses and influenza for the last 3.5 years. So I think about these things quite a lot, and I directly work with all of the pathogenic human coronaviruses and some of the low pathogenic coronaviruses. So the next comment that came up that I'm using as my springboard into the next stuff I want to talk about came from Michael Kukin, which reads, if a lab wanted to experiment on RBD amino acids to test the 6-chain combinations, how long would the experiments take to test one? Are labs able to test multiple at a time? And if so, how many at a time would a lab such as one your size reasonably be able to test? I'm wondering how unreasonably vast the scale would be to experimentally test all 6 million and the scale of resources it would require. So my reason for using this comment is that it will directly lead me talking about a paper that I read today that was published today, and for how that's influenced my thinking on everything and led to me making this video this way. So just as a quick reminder, the RBD, the receptor binding domain, is essential for how a virus infects cells. So on the surface of coronaviruses, you have the spike protein, which sticks out, and that binds to the receptor ACE2 on the surface of human cells. The lock and key is the metaphor I always use. The receptor binding domain is the most important part of spike for binding to the receptor, as the name implies. With the lock and key analogy, it's kind of like the teeth of the key. So if they're not the right shape, you're not going to be able to unlock that lock. So the RBD is absolutely essential. And the discussion I was having in the previous video is that the SARS1 spike protein, the RBD, there's 6 amino acids that are very essential or important to the binding to ACE2, so the receptor binding domain. And 5 of those 6 are very, very different are just different in SARS2. If you were to computationally model a sequence of 6 amino acids and how that would interact with ACE2, which you can do, the predictions would come up with the fact that the SARS2 RBD is not good for binding to human ACE2. So my argument was around the fact that you do the computational modeling instead of doing all the combinations of 6 amino acids at that site, because to go and do that, you'd have to make viruses or spike proteins that carry every different combination. Now, the way I was thinking that may be done and why I thought it's unlikely would be that you'd have to produce spike proteins that have, as I say, all of the different combinations. You'd probably use a spike that already infects human cells if you were doing this to look at human cells. So it would really closely resemble one of the human coronaviruses we already know about, because other parts of spike are important for infection of cells beyond the RBD. So you'd have a backbone of a human coronavirus, and then you'd play around with all of these 6 receptor binding domains, amino acids. So then my argument went along the lines of you'd end up having to make 6 million different combinations and have to make viruses that carry those. Now, the way that would be done, if it were to be done, it could be done. You could buy the DNA that produces all these different spikes, and you could do it. It'd be a huge labor to do, but it could be done. And then you'd make a virus that has each of those spikes on a viral core of something that doesn't infect or doesn't cause disease or can't cause disease. So this is something called pseudotyping, or making a pseudotype virus. So you take something that doesn't cause disease in humans, for example, a virus called VSV, vesicular stomatitis virus. It's a virus of cows. That virus can infect cells in the lab, but it doesn't cause any disease in humans. What you can do is you can take the core part of that virus and then put on any kind of different spike protein or glycoprotein. So spike is coronaviruses, glycoproteins is the overarching term for these type of proteins. Because that way you can then have this core that won't cause disease, and you can test the determinants on the spike that allow it to infect cells. The way you make this whole system even safer for potential release from a lab, and I'm going to be coming back to this shortly, which is why I'm going into detail on it, is that you take out a huge amount of the genes within this core and you replace things with fluorescent proteins, for example, or other types of markers, so you can easily see if it's infecting cells. So the experiment idea is you want to see whether this spike versus another type of spike can allow that virus to infect cells better. And the way you can do is that your core produces a fluorescent protein, so you just look for the cells that are glowing. And when you do that, you can take out all the kind of genes that may allow you to make new viruses. So you just look at one hit infection. You don't produce any more virus. So that's the way you can do all this kind of stuff safely. You use a virus that doesn't infect humans or doesn't cause disease in humans, but can infect cells in the lab, make it fluorescent, and make sure it's a one-hit virus. It just goes in and nothing else comes out of those cells, but they glow green. So where my head was at in the last video was that you'd end up having to make this huge number of viruses and you'd have to infect cells and do this laborious thing. And I thought, therefore, it's more likely that a computational approach would have been used at the front end, which would have ruled out the SARS2 spike. And then you'd have just tested those if you wanted to do this. However, this leads me into the paper I read today. So the paper's up on bioRxiv, and I'm going to link to it down in the description for this video. And what they've done in this paper is they've looked at every possible combination of the SARS2 RBD to see which ones influence binding to ACE2. Can you get better binding, or is SARS2 the optimal binding? Which, being that we've only had SARS2 around for a few months, means they've done that in a few months. So it's a lot more feasible than I'd realized. So I therefore have to admit a gap in my knowledge. I realized I did know this system existed. I'd just forgotten about it because I hadn't read the papers or thought about it for a long time. But anyway, this paper is a wonderful paper. If any of the authors are watching, it's a great paper, and I'll explain why it's important they've done it. But to explain what they've done, in the paper, as I say, they've looked at every possible combination of the SARS2 RBD amino acids to see how they influence ACE2 binding. The way they did this was using yeast. So it's possible with yeast to make them express the spike protein on their surface. So instead of having your virus, I'm going to do this for a yeast, you end up with the spike of a coronavirus on the surface of the yeast. And then you can look to see if those yeast will stick to ACE2 proteins. So then they used this clever technique involving PCR, which copies DNA, which is a degenerate PCR. So you just make a load of mutations. You make this huge mutational library that will have every combination of the DNA sequence to make spike amino acid combinations, and they barcode all of those so you can tell which one's which. So they made this huge library, mutational library of all the different combinations, and then they individually expressed those into yeast to see which ones would bind best to ACE2. So they did this. They obviously selected for the yeast that could bind best and looked to see how well different combinations of the RBD bound to ACE2. What they found was that the SARS2 site is pretty optimal. It's not the strongest binder. There are stronger combinations of these amino acids, but it's right up there. There's many combinations that don't bind as well as SARS2 spike, and there's many that don't even produce a functional protein. So all of these mutations that you can look at, they don't necessarily even produce a functional protein that goes to the surface of the yeast. So there's not actually 6 million combinations. I got that one wrong when I was describing it last time. There's fewer combinations because they don't all produce a functional protein. Anyway, so why that's important is that they've been able to look at all these different combinations and find really strong ACE2 binding sequences. And I will come into why that's important with regards to the idea of the lab hypothesis if you've not already jumped to that. But before I do that, I want to say why that's been important to do for SARS2. So the RBD, the receptor binding domain, is one of the main targets for vaccination. So when we vaccinate people with any of the different approaches, 120 different vaccines or whatever it is now that are being trialed, that's trying to produce antibodies that will target the spike protein of SARS2 to stop it interacting with ACE2 and therefore stop it infecting cells. A great target for those antibodies is the receptor binding domain. If those antibodies come in and bind to that receptor binding domain, that's a great way to stop it binding to ACE2. Now, if the receptor binding domain of SARS-2 were to mutate, that would stop the antibody binding as well, and then it can go on and infect cells again. This is what happens with influenza, I should say, and which is why we need seasonal vaccinations against influenza, because the HA, the equivalent of spike, mutates every year, and it escapes the antibodies that were produced from the vaccination the previous year. So if we know all of the combinations that there can be for the SARS2 receptor binding domain and how they influence binding to ACE2, we can look to see if any of those are emerging in people who are being infected with this virus. So viruses are constantly being sequenced, they're infecting people, and we can see if there's any changes going on. And if we see that there are changes going on, we know that we're going to need to change our vaccine. So it's really important work, and it's great work that they've done. But obviously now we can go into the discussion of whether this virus came from a lab, because if similar research had been done and the top candidates for receptor binding were found in, I'll say, the lab in China, because that's where the fingers are being pointed. If that research was done and they were looking to see experimentally whether those different receptor binding domain sequences had an impact, this is the kind of way they could have found it. It's a lot easier than the way I realized it could have been done. Now, what was done in the paper I looked at today was to actually make pseudotyped viruses, not just the yeast, but they made pseudotyped viruses with their top hits, and looked to see if it really worked when you had a full spike as well, so on a virus particle. But it was in this pseudotype system, so that kind of system where it's a lot safer, that you don't have all the problems that if it does get out of a lab, it's going to go in and infect people. But obviously, it could be done in a virus that could infect people. So here I will say that it's possible that that was done. That research could have been done. They could have been looking to see if there are different sequences of the receptor binding domain that allow spike to bind better to ACE2. And then to come back to the furin cleavage site, obviously the furin cleavage site, as I was saying, could impact the transmissibility. So then you do the combination. You'd have the receptor binding domain and you'd have the furin cleavage site, and see how that influences the infection of your pseudotyped viruses to see which ones are best. Now, if you were doing that, you'd, as I sort of made the argument before, you'd probably be working on the backbone of a virus that's already known to infect humans, if you were just looking to see what kind of sequences. But alternatively, you may do it on the backbone of a bat spike sequence to see if those mutations could go into a bat spike and allow it to infect human cells. So potentially that was done, and it should have been done in a pseudotyped system if it was being done, so that it's safe. But you can, of course, make the argument that it was done in a full virus that came from a bat. That can't be ruled out. People may have done those experiments, which, if you want, which may have been done. But here's where the jump has to be made. So right now, to the best of any knowledge that is openly available, the closest relative virus of SARS2 is the RATG13 2013 virus, which is distinct enough from SARS2 that they've probably had 25 to 65 years of evolution. So unless there's a virus that is 99.99% similar to SARS2, that's only really different in certain parts of the spike, maybe some other parts if you've done some passage and those kind of things, that's where I start to come to the idea that you have to make the accusation that there's a cover-up, which I'm not comfortable doing, as I said at the top. But I will concede you can make these arguments. So it is possible that people were working with these kind of experiments in the lab, looking to see the determinants of how a bat virus could infect a human cell, and that there could have been an accidental release. If you just go and Google, there's lots of examples of accidental releases of viruses. It looks like most are from America when you read it, but that's because we typically in America disclose that information better than other places. So you can very easily arrive at a place where you can make a compelling argument for the fact this virus would have been studied, looking to see the best binding to ACE2, the kind of things we should be worried about, and whether the furin cleavage site has an impact on infection of cells in the lab. The jump, I should miss a finger, I guess. The jump comes to in my view as the virologist, that there has to be a virus that we don't know exists, and therefore you have to accuse of a cover-up. So this is why I land on the thought, the idea, the argument that this came from nature. I think with all the genetic diversity out there, the fact that recombination can occur at the fusion site, the fact that these furin cleavage sites exist, the fact that we found the same receptor binding domain sequence in viruses and pangolins, I think all of that points towards it evolved in nature, that it can evolve in nature, and that it did. An argument that I've also heard made, so I've listened to podcasts, and I don't want to name names, I don't want to throw accusations, but I've listened to podcasts discussing this, I've read things online, and one of the things that keeps coming up is sort of around the fact that it's a huge coincidence that this virus would emerge at the Wuhan seafood market, right near the Wuhan Institute for Virology, where this kind of research is being done. For that, my argument is that I'm pretty sure that we're going to find good evidence that there were earlier cases further away from Wuhan, where it already seems that the seafood market wasn't where this virus emerged. There's evidence now of cases back in November. Some predictions on evolutionary timelines predict September may have been the earliest infections. Wuhan seafood market seems to just be a super spreader event. So one person went in and infected a lot of people. Something that we see happen with coronaviruses. We don't really know why, but it happens. And so it is a coincidence. I will say it is an amazing coincidence, but I still strongly feel that this virus emerged in nature. We've seen it happen multiple times. We've seen it with SARS. We've seen it with MERS. We've seen it with H1N1 in 2019. Every previous virus we've seen has come from nature. And so with that and the genetic diversity, I think for me, it's a stronger argument that this virus was something from nature. But as I say, if I, to myself or to all of you in this video, make the arguments for the fact it could have been something produced in a lab, I do end up at a place where it could have been something produced in a lab. But I'm not throwing those accusations out. I'm just trying to have the open, honest discussion about it. Ultimately, I think we'll eventually find this out. I think over time, as that coronaviruses get studied more, we will find that this virus was something in nature, or that we'll find much closer relatives to this virus in nature. It took a long time to find that SARS-1 had this transmission route from bats to palm civet cats to humans through a wet food market. So I think that's what will happen with this. That's my speculative hypothesis as a scientist. Anyway, as I said at the top, part of this channel is me talking about coronaviruses, tackling issues regarding this pandemic, and admitting when I'm wrong, because there's new information that comes all the time. And I have no doubt I will come back to this topic in future videos, because I'm sure there'll be new things that come out that change the way I think, and I will keep approaching it. But hopefully I've made it clear that I think both sides can be argued, and I am landing on the side of it being a natural virus. Anyway, another long video, possibly a bit convoluted in my explanations, possibly again very heavy on the science. It's a bit convoluted because my brain's a bit convoluted on it all as well. But hopefully people were able to follow. If not, please leave questions and comments, ask me to clarify on things, and again, leave questions and comments, because what I may well do is another video based on those questions and comments as I had planned last time. Otherwise, I'll go into new videos and talk about new things that are coming out, talk about some new vaccine strategies, possibly talk about monoclonal antibodies. A company we've worked with published a paper in Science on Monday, and it's going into clinical trials for monoclonal antibody therapy, and I've not even talked about that once in the video, so I'll probably be doing that soon. But anyway, as I say, please leave questions, comments, give me feedback on these videos. I know they're currently a lot longer than I want them to be, but this is a big topic that requires a lot of talking. As always, if you're enjoying the videos, please subscribe and drop a like and hit the bell so you get the notifications when I post new videos, because it's a bit erratic at the minute. And as always, please stay safe, wash your hands, and keep calm and carry on. We will get through this pandemic.